is we're going to use Skittles to kind of simulate what we did in our gene transformation. And that way, hopefully you'll understand all the different processes that we took part here and why our results, what we learned from our results. Whatever I just said. Okay, now, this yellow Skittles are going to represent regular E. coli bacteria. Bacteria with not added genes, they're just regular E. coli bacteria. The green Skittles are going to represent bacteria that got the plasmid, which means they would have gotten the what? Genes for bioluminescence and
ampicillin.
The big question I want to look at here is why do we need the ampicillin? Why do we need that gene for resistance to ampicillin? The thing is, our goal is to get bacteria that can give off light, that can bioluminesce. Seems like all we need to do is give them that one gene. We're going to see why that second gene was so important to add to this. It was actually very smart. The scientists who came up with this technique, it was sheer genius in order to do this. So what we did is we took some bacteria and we put them in control tubes, cold calcium chloride, in plastic tubes, cold calcium chloride. We heat shocked them, we put them back on ice, we gave them some nutrients, gave them a little love, and then we poured them on the plates. And Marta went crazy with the marbles and things like that, but we spread them around, and then we incubated them, correct? All right, so these are the plates. We're pretending these are the plates that had agar on them. So let's start with the control, which means these are the bacteria that did not what? Receive the ampicillin, the plasmids, right? So we're going to put them on regular nutrient agar, regular nutrients, and we're going to put them on agar with ampicillin. All right? Now, the next day, what was our result here? It was totally covered in bacteria. Why?
Because agar has nutrients.
Agar has the nutrients in it. So when they reproduce, one handful of Skittles becomes 2 handfuls of Skittles. 2 handfuls of Skittles become 4 handfuls of Skittles every time you reproduce, and so on. And ultimately, this entire plate gets covered with bacteria. That was our result. That's what we thought would happen, and that's what did happen. Why was that important? Because it told us what? We didn't harm the bacteria in this process. Very good. Meanwhile, what would the ampicillin do to these bacteria?
It would destroy the bacteria.
It would kill them because ampicillin's a what?
Antibiotic.
Antibiotic, and that would destroy these bacteria because they don't have the resistance for it. They have not gotten the genes for it. So that's what we ended up with that plate. There was no bacterial growth in it, and it told us our ampicillin was what? Working and effective and good. Okay? All right. So now let's focus on the experimental part. I'm going to pour these out here and get these out of the way. Now we're focusing on the experimental plates, the ones that possibly could have gotten the genes for
bioluminescence and the resistance to ampicillin.
Good. So that was our other tube. All right? We put them in cold calcium chloride, just like the control group. We heat shocked them. We put them back on ice. We gave them a little nutrients, gave them a little love, poured them on the plates, and then we poured them on agar and also agar with ampicillin. So, how many bacteria got the plasmid here? 1. How many didn't? A lot more. And then over here. How many got the plasmid?
1.
How many didn't? A lot more. So let's focus on this plate now. This is regular LB agar, regular nutrient agar. We know that we have 1 bacteria cell with the plasmid. These did not. So now we're going to reproduce them. 1 cell will become 2. A handful of these will become another handful. 2 handfuls, right? 2 handfuls will become 4. That's 3. 4. 2 cells will become 4. How many handfuls I have? 4? 4 handfuls will become
5.
Nope, they reproduce. They double.
Exponential.
They double. Nope, this isn't funny. This is serious. All right? So 4 handfuls will become 8. All right? So I'm up to 4, 5, 6, 7, 8. Meanwhile, the 4 cells that got the plasmid are going to reproduce, and I'll have how many? 8. What do you see happening here?
There are more.
Go ahead, Alex.
There are more bacteria without the plasmids, and there are less.
Well, we started like that too. Well, what's happening as we keep reproducing?
There's more and
The
more.
difference between the bacteria that doesn't have the plasmids and the bacteria that do is growing. So
Is
we're getting-
exactly, all right? The gap is greater. Now we have, at first we had, I don't know, maybe 5 or 6 more that didn't get the plasmid. Now we have hundreds more that didn't get the plasmid than did. So how many handfuls were there? And eventually, these guys are going to multiply, what, I have 8 handfuls? Then I'd have 8 more handfuls. I'd have a lot more of these. And then these guys would reproduce again. And I'd have maybe another 4. And in the end, what's happening here? What's happening to the green ones?
The difference.
They're overwhelmed. Good. They're being overwhelmed by all the others that
Don't have
did not have the plasmids. They're also isolated from one another. This is going to become important in a lesson on Monday. Do you guys want to see what's happening here?
Yeah.
Now, meanwhile, let's look at the really important plate. The one that had ampicillin and agar. With the ampicillin, what would happen to the bacteria that did not get the plasmid?
They would all
They would all die. That would leave us with what? Only
the bacteria.
this
that got the
bacteria that what? And what is that bacterium able to do? I know it can reproduce. It can what? It can resist ampicillin, but more importantly, our goal was to get bacteria that can what? Glow. That one can glow. 1 becomes 2 become 4. They divide again. 4 become 8.
So is that why we don't see like as much glowing because there's
In this one?
Yeah.
Yes. Or maybe none at all. That was the point. That's good, Glenn. That's exactly where I'm going with this. Meanwhile, right here, 8 become 16. That looks about right. You get the idea. You see what's happening here. The only cells that are able to survive in this plate are what?
Those that have plasmids.
Now, I want you to think of how smart this was. Why are the only ones, let me say this again. Why do we only have cells that can glow on this plate?
To isolate.
Because why? Cooler?
Ones died
But why did they die and these
because
didn't?
they couldn't resist ampicillin
Why could these?
because they had the genes to resist.
Thank you. The reason these guys are able to exist and glow is because they also have the gene to resist ampicillin. Now, how smart is that? I want you to think about this a minute. If I wanted to separate out the bacteria from those that didn't get the gene, the ones that did from those that didn't, could I do this?
Yes.
I mean, no, wait a minute. I want you to think about it. Could I physically, could I take tweezers and actually
No.
pull bacteria cells out? It would be impossible. So think of how smart the scientists who invented this procedure were. They said, we need a way to separate and isolate and filter out the ones that got the plasmid from those that didn't. How do we do it? We kill the ones that didn't.
have the plasmids.
But to make sure these guys survive, we give them the resistance to the ampicillin. Pretty cool, right? All right. Thank you very much, guys.
It's on YouTube.
It's been a great job.